Hi everyone, I am Dr. Devya Boha, and in this video lecture I will be discussing the role of UHPLC in pharmaceutical analysis. UHPLC has wider application for the pharmaceutical analysis because the separation efficiency in UHPLC is more as compared to the HPLC, whereas the resolution power would also be more for UHPLC as compared to HPLC. UHPLC has increased sample throughput, which means that in less span of time you can analyze more number of samples, and the analysis time is also lesser. So in different ways, UHPLC technique is more superior as compared to normal or conventional HPLC for the pharmaceutical analysis. And the very important point is that UHPLC is very helpful in separating closely eluting compounds. Now, closely eluting compounds means the retention time of the compounds would be very nearer to each other. So, there would be very less time gap between their elution, right? So, due to this, what will happen in the chromatogram? The peaks of the closely eluting compounds would be nearer to each other. So since in UHPLC resolution power is high, the peaks won't get overlapped with each other even though they are close to each other. Whereas if we talk about conventional HPLC, where the resolution power is lesser than UHPLC, if you try to elute or if you try to separate closely eluting compounds, then it would be difficult to do so. Why? Because in the chromatogram, for HPLC technique, closely eluting compounds peak would be closer enough and they might overlap with each other. Okay, and we don't want that the peaks get overlapped with each other at the base. Okay, so with the help of UHPLC, separation of closely eluting compounds is more easier and it is more preferred as compared to conventional HPLC technique. So now let us take some of the examples of the drugs which are being analyzed by HPLC and UHPLC. So here we'll be taking the example of three drugs. Okay, so first drug is drug A, or you can call it as compound A. Second one is drug compound B, and the third one would be drug compound C. Right, so the same drug, when it is being analyzed by conventional HPLC and UHPLC, what are the differences in the parameters which has been set into the instrument? Let us see that. So let us start with the drug A, right? So the column which is used in HPLC for the separation of drug A, suppose say it is Phenomenex Aqua C18 column. So over here, Phenomenex is the name of the company which provides the column, whereas the Aqua is the series of the column, I mean the name of the series, okay, and C18 is the column type, right? So C18 columns are known as octadecyl silane columns, okay. So over here, 18 represents that in the molecule which is being used as a stationary phase would be having long chain of carbon, and there would be 18 carbons in that long chain, okay, and these carbons would be bonded to the silica particles. Okay, so that's why it is referred to as octadecyl silane columns. Okay, octadecyl because 18 carbon atoms would be there in the molecule, and silane because this carbon atoms would be bonded to the silica particles. And this type of columns would be the reversed phase columns, and the stationary phase, which is C18, would be non-polar in nature, and that's why it is used for the separation of the polar compounds, right? And the mobile phase, which would be used for the separation, would also be polar. So most of the drugs are organic compounds, and we know that organic compounds are polar in nature, right? So they can be well separated by using the reverse phase chromatographic technique, where the stationary phase would be non-polar and the mobile phase, which would be used for the separation, would be polar in nature. Now over here 3 micrometer is the particle size of the stationary phase, whereas these are the column dimensions. The length of the column would be 150 mm, okay, whereas the internal diameter of the column would be 4.6 mm. So this is the column which is being used for the separation of compound A, okay, which is drug A, okay. Now let us see which column is used in UHPLC for the separation of the same compound. So in UHPLC, water's equity BEH C18 shielded RP18 column is being used. Okay. Now waters is again a company like that of a Phenomenex. Equity is the range of the columns which is being provided by waters, right? And BEH means ethylene bridge hybrid. So this type of columns which are used in UHPLC are hybrid columns, okay. Now again, this is a C18 column, okay, and RP over here represents reverse phase columns, okay, and C18 again would represent the same thing, that there would be a long chain of carbon where number of carbon atoms would be 18 and they would be attached, or you can say bonded to the silica particles. Okay, so it's again a reverse phase column which could be used for the separation of organic compounds, or you can say polar compounds such as drugs. Okay, over here the particle size of the stationary phase is 1.7 micrometer, and if you see the column dimensions, then the column is 20 mm long and 1 mm would be the internal diameter of the column. So if we compare the parameters of both of this type of columns, then you can see that the particle size of the stationary phase in HPLC columns are more as compared to the UHPLC particle size, right? So this we have already discussed in the previous lecture, that the particle size of the stationary phase, or you can say the column material, would be very less in UHPLC, okay. And even the internal diameter of the column is lesser in UHPLC. So the columns which are used in UHPLC would be more narrower as compared to the HPLC columns, and even the length of the columns would be lesser. You can see over here there is a huge difference in the length of the column. Over here it is 1020 mm column, and over here it is 150 mm column. Okay, now for the same separation you can use another column, okay, that is Phenomenex Luna. So over here the name of the company is Phenomenex. Luna is the range of the series name, or you can say range name of the column, right? So Phenomenex Aqua, Phenomenex Luna, similarly Phenomenex Synergy. So different series of columns, or you can say different range of the columns are there which are being provided by Phenomenex company. Okay, so Phenomenex Luna C18 to HST column is being used. Over here HST means high speed technology. Okay, so again it is a C18 column. It is a reverse phase column where the stationary phase would be non-polar, okay, and the molecules would be having long chain of carbon atoms where the number of carbon atoms would be 18 and the carbon atoms would be bonded to the silica particles. And over here the size of the particles of the stationary phase is 2.5 micrometer. So it is anyway lesser than the particle size of the stationary phase which is used in HPLC columns, right? So you can see that if we see the column dimensions, then it would be always less in UHPLC as compared to HPLC, and even the particle size of the stationary phase would be lesser in UHPLC as compared to the HPLC. Now let us see the flow rate. Okay, so this is the flow rate of the mobile phase. So it would be in ml per minute. So the flow rate in HPLC would be generally set 1 ml per minute for the separation of this particular drug, whereas in UHPLC it would be 0.4 ml per minute. So you can see over here the flow rate would be lesser in UHPLC as compared to HPLC. So the consumption of the solvent would be lesser in UHPLC and the overall cost for the analysis would decrease due to this. If you see the injection volume of the sample, then it would be 10 microliter in HPLC and it would be 5 microliter in UHPLC. So if you are having a less amount of the sample, then UHPLC could be preferred more over HPLC technique. If you see the temperature, then the temperature would be around 35 degrees Celsius in HPLC and it would be 50 degrees Celsius for UHPLC. Runtime is 40 minutes for HPLC and it is 4 minutes for UHPLC. So you can see over here there is a drastic change in the runtime. It is very less in UHPLC and it is 10 folds lesser in UHPLC when you compare it with that of the HPLC technique. So these are the parameters which were set for the compound drug A separation. Now let us take another example for a drug B. It is another type of drug. Okay, so again it is a organic molecule, so it would be polar in nature. So again you have to use a reverse phase column. So the column which is used for HPLC analysis of this particular drug, the column used is Waters Sunfire, right? So Waters again is a company and Sunfire is the name of the series. Okay, over here you have seen that water equity columns are available, so Water Sunfire columns are also available. So again it is a C18 column. Now you know what is C18, right? It is having 3 micrometer particle size, okay, of the stationary phase, and the column length is 150 mm and the column internal diameter is 4.6 mm. Over here, Waters Equity BEH column C18 shield RP18 can be used. 1.7 micrometer is the particle size. 1 mm internal diameter would be there and 20 mm long column would be there. Phenomenex Luna C18 to HST 2.5 micrometer column can be used in UHPLC for the separation of compound B, right? So this both our columns are similar. I mean this the same column is used for the separation of drug compound A and drug compound B using UHPLC. Now if you see the flow rate, over here the flow rate is 1 ml per minute, over here it is 0.4 ml per minute, so it is less in UHPLC. Injection volume is 10 microliter here and it is 2 microliter here, so again injection volume is lesser in UHPLC. And we have already studied in the previous lecture that the maximum injection volume which you can take for UHPLC is 8 microliter, right? And the temperature is 30 degrees Celsius for HPLC and 50 degrees Celsius for UHPLC. Temperature would be slightly higher for UHPLC analysis. Runtime is 35 minutes and over here it is 8.5 minutes. So you can see that analysis could be fastly completed. The analysis would be speedier in UHPLC as compared to the HPLC with higher resolution. Now comes the compound C, where the Phenomenex Synergy polar RP columns are being used. Okay, so again Phenomenex is a company, Synergy is a series or the range of the columns which are being provided by Phenomenex. Polar RP means RP over here refers to the reverse phase and polar over here represents that this column is able to separate the polar compounds. Okay, so that's why it is referred to as polar RP columns, that is polar reverse phase columns, wherein the mobile phase which could be used would be polar in nature and the stationary phase would be non-polar, and again it could be used for the separation of drugs which would be mainly polar compounds. Okay, so Phenomenex Synergy polar RP columns are sometimes even referred to as ether bridged phenyl columns. Okay, ether bridged phenyl columns. Over here in this figure you can see that this is the phenyl ring, this is the ether group, okay, and it is attached again to the carbon chain, and over here at the end the carbon would be bonded to the silica particles. And this over here AQ represents the aqueous form because this type of columns are hydrophilic type. Okay, so they attract the water molecules, so that's why AQ, that is the water molecule, is being attached to the silica particle. Okay, so over here again I'm repeating, this is the phenyl group, this is the ether functional group. Okay, and then there would be an chain of carbon atom wherein the carbon at the end would be attached to the silica particles. So this is a Phenomenex Synergy polar RP columns, right? and it is sometimes even referred to as ether linked phenyl columns, right? Because through ether the phenyl rings are linked with the carbon chain, and then the carbon chain would be bonded or linked with the silica particles. So over here the stationary phase particles, or you can say these particles would be having particle size 4 micrometer. Okay, and the length of the column would be 50 mm and the internal diameter would be 4.6 mm. So for again, if you go for UHPLC for the separation of compound C, then water's equity BEH column is being used. Okay, right, and the particle size we know in BEH columns would be very less. It would be approximately around 1.7 micrometer. That would be eventually very less than the Phenomenex energy columns. Okay, and the flow rate which are being set over here is 0.8 ml per minute. Over here it would be half of that, that is 0.4 ml per minute. Injection volume is 20 microliter over here, wherein in UHPLC it would be 5 microliter. Temperature, if we see over here, it would be 35 degrees Celsius, wherein over here it would be 50 degrees Celsius. Runtime is 40 minutes in HPLC. It is 19 minutes in UHPLC. Right. So if we compare all of the properties, or you can say all of the parameters which are being set inside the instrument, then there would be a huge variation if we talk about conventional HPLC and UHPLC. And through various things we can see that UHPLC is always superior as compared to the conventional HPLC technique. So one of the major difference between HPLC and UHPLC method is the column dimension. We know that in UHPLC the length of the column would be lesser and even the columns would be more narrower, which means that the internal diameter of the UHPLC columns would be lesser. And even the particle size, or you can say particle diameter would be lesser in UHPLC columns as compared to the HPLC columns. So when the columns are having shorter length, it would reduce the back pressure. Okay, you should understand what is back pressure, because over here we know that the instrument is being operated under high pressure, and since the column which are mostly used would be the completely packed columns, and the particle size of the stationary phase is also very small. It would be in micrometer, right? So sometimes, even if the mobile phase is being passed through the column at higher pressure, there would be an issue of the backfiring of the mobile phase. Why? Because it is difficult for the sample, or you can say the mobile phase, to pass through the small size stationary phase particles, right? So there would be an issue of the back pressure. Okay, the mobile phase which consists of the sample might be thrown back, right, in the reverse direction. But if the length of the columns are shorter, then this back pressure would be lesser, or you can reduce it. Wherein if the columns are narrower in size, then we can minimize the frictional heating effect. Now, what is frictional heating effect? That we have already studied in the previous lecture, right? Frictional heating arises due to the friction of the mobile phase particles to that of the stationary phase particles, right? They due to the friction of the particles of the stationary phase and the mobile phase, heating is being produced, and that could be minimized by narrowing the size of the column, because we know that the inside the column the temperature would be more at the center as compared to the wall, right? So if you decrease the diameter, internal diameter of the column, then this frictional heating could be prevented. Now, in UHPLC, we know that the flow rates are very lesser and even the sample injection volume would be lesser. So this will help in lowering the solvent consumption and its disposal. And since the injection volumes are lesser, sample conservation is possible, right? Because less amount of the sample is used for the analysis. So in the cases where the drugs are very costlier, UHPLC would be more beneficial because less amount of the sample is required for the analysis. And in some cases, what happens that you will be having very less amount of the sample, right? So it would be difficult to go for HPLC wherein more amount of the sample injection volume is required. So in such cases also UHPLC would be preferred as compared to the HPLC. Now the next thing is the runtime. We have already seen in the previous three tables that the runtime is very lesser in UHPLC. Over here it is 10 folds lesser, right? Wherein over here it is 4 folds lesser, and over here it is 2 folds lesser as compared to the HPLC. So runtimes are always lesser in UHPLC, and due to this the sample throughput increases, which means that in less time you can analyze more number of samples. So this technique, that is UHPLC, is capable of achieving higher peak capacity. We know that over here in UHPLC band broadening would be lesser. So the peaks would be more narrower, the peaks would be more sharper, okay, and the resolution power would be more. The technique is more speedier, right, and the sensitivity is more, and it would be able to analyze the samples which is having very lesser concentration. So due to all of these reasons, UHPLC has increasingly used in pharmaceutical industries from drug discovery to drug development. Now we know that all of the pharmaceutical samples are very complex drug molecules, right? And there would be a number of samples which are needed to be analyzed. So number of samples could be analyzed in a short span of time with better separation, with higher resolution and higher sensitivity, which could be achieved by using UHPLC, right? So finally, we can conclude that for the pharmaceutical products, it would be easier to analyze the compounds using UHPLC as compared to the HPLC. Now let us see the chromatograms of the drug products which have been obtained from UHPLC method and HPLC method. So these are the preparative analysis graphs, right? You know that what is preparative methods, right? Where this we have already studied when we were studying the applications of SFC, right? So preparative methods or preparative instruments are those instruments through which once the compounds are being separated at the end, you can separate the individual compounds separately. Okay, so this is the chromatogram for the analysis of a drug compound which is being done using conventional HPLC, and this is for water equity UHPLC analysis, right? So in this you can see that the peaks are quite broader as compared to the peaks which are given in this chromatogram, right? The width of the peak is more in this chromatogram as compared to this. So naturally the band broadening is more, right? Peaks are wider, and you can see that peaks are overlapping with each other at the baseline, right? You can see over here there are two peaks and they are overlapping at the baseline. Over here also the peaks are quite overlapping at the end. Okay, and the runtime is higher. Over here the runtime is 10 minutes, but if you see over here it is just 1.5 minutes. Okay, but the if you analyze the same drug using UHPLC, then you can see that the peaks are quite sharper. Okay, the width of the peak is lesser. Okay, the bandwidth is lesser. Okay, and the separation is found to be better over here as compared to what is being achieved in this particular chromatogram. Okay, and you can see the specific peaks over here, right, without overlapping at the base. The same peaks you can see over here, they are overlapping at the base. Over here there is no overlapping. You can clearly see two individual peaks over here, whereas if we see these two peaks, then it has been overlapped a lot. If you see over here, the overlapping is just, I mean, it is very less. right? And even the runtime is very less in UHPLC. So you can see the compare both the graphs, and you can see that the resolution is more in UHPLC. Over here, RS means the peak resolution, right? And it is said that if you want to go for the separation of a pharmaceutical products, then the resolution should be always higher than 1.5, right? So here you can see that real resolution is very less, it is 0.5. So that's why the peaks are overlapping with each other, right, at the base. Here resolution is 1.53, so that is quite okay. Here the resolution is 1.44, that is nearer to 1.5, so that is okay. And here the resolution is very good between this two peak, that is 2.84, right? So you can see that resolution power is more in UHPLC as compared to the HPLC. And now you can make out that if the resolution is more, then the peaks would be further apart from each other and they won't overlap with each other, right? If you see these two peaks in this graph, then resolution is just 1.5. So at the base, the peak is slightly overlapping. Over here, since the resolution is more, that is 2.84. So there is a huge gap between the two peaks and they are well separated from each other, right? And make sure that these chromatograms are for the same drug compound, wherein this peak is this one and this peak is for this particular compound, right? So if you compare the graph, then you can easily found that in UHPLC the band broadening would be lesser, resolution would be more, and separation would be done in a proper way. There would be less time consumption, which means that analysis would be done faster. So the solvent consumption, that is the mobile phase consumption, would be lesser. So economically the UHPLC process would be more better, right? Because it would be more economical, less costlier, right? So there are various reasons through which UHPLC is superior as compared to the HPLC technique. So that's it for this particular topic, and with this, we finish the applications of UHPLC for pharmaceutical analysis. Thank you.